Our next speaker is Dr. Ola Gutzeit. Ola is a former research fellow at the Wyss Institute at Harvard University, and currently she is an attending physician, IVF unit, Rambam Medical Center, and she is also a researcher at the Clinical Research Institute at Rambam. So, Ola.
Hi, thank you so much. Okay, so about 3 years ago, a couple came to my clinic, and they came because they were trying to conceive without success for over a year. So we did all the known evaluating tests, and unfortunately, we couldn't find the reason for their infertility. Despite the fact that we didn't know the underlying cause of their problem, we could offer them a treatment. So after a few cycles of fertilization, we succeeded and we reached a pregnancy. However, 24 weeks later, we received them to the delivery room with signs and symptoms of preterm birth, and they delivered twins that none of them have survived. So infertility affects 186 million couples all over the world. And every fourth couple that come into my clinic will be diagnosed with unexplained infertility. Preterm birth is #1 cause of death in children under the age of 5. So we have a very significant gap in women's health. There's many things that we do not understand, and there's many reasons for this. So first, women were excluded from clinical trials for many years by the FDA. Second, the models that we are using are insufficient. The animal models are not good enough, and there is a significant gap of knowledge. So with the understanding that we need a significant breakthrough in the field, and with the patients in my heart, I moved to this institute at Harvard. So the Wyss is an institute for translational science that brings together scientists, engineers, clinicians, and trying to find translational solutions for medical care. So at Wyss, I joined a group that we use organ-on-chip technology. So what is an organ-on-chip? The organ-on-chip is a 3D culture that we can culture a few cell types with a flow and a stretch. Basically, we are recapitulating a functional unit of an organ. So we have developed a vagina on a chip, a cervix on a chip, endometrium. We're still work on the endometrium, and we are at the final stages of developing fallopian tube on a chip. So why using a chip is a good solution for women's health? So first, we can do patient-based, individualized medicine. So we can use the primary cells for individual patients and recapitulate their biology. We can study dysbiosis and we can study microbiome, which is very complicated process within women due to the fact, within the women reproductive tract, due to the fact that it changes very significantly throughout the menstrual cycle. We can understand how the host is responding to the microbiome, how this host is responding to different treatments, and we can look for novel markers to diagnose our diseases. So we have used control chips, which just have the functional units of the cells. We have a good microbiome, a healthy microbiome environment, which is basically lactobacilli based, and we have a bad and bright microbiome, which consists of a bacteria that represent bacterial vaginosis. So dysbiosis is associated with the most significant problems in women's health. So it is very significantly associated with preterm birth. It is very significantly associated with dysbiosis. However, we don't know what is the underlying fact, and this is where this study is coming from. So just a brief overview. So the first thing I did, so basically I connected the vagina on chip and the cervix on chip, and we looked at how the microbiome is changing and what is the effect of the cervical secretion of the microbiome within the lower reproductive tract. And we showed for the first time that the cervical mucus within the female reproductive tract is basically regulating the microbiome. So then I'm a reproductive endocrinologist, and I want to see whether we have an effect on how can we affect fertility. So there is significant association between dysbiosis and infertility. However, we don't know what is the underlying cause. And due to the fact that this is not something that we are screening, this is not something that we are treating, and when we are looking for unexplained fertility, the microbiome within the lower reproductive tract is not one of the tests that we will perform, and this is not a treatment that we will offer. So we use the cervical chip as a model, and we treat it with dysbiosis, and then we put a sperm on the lower reproductive tract. And then the first thing we saw when we compare control to healthy and dysbiotic conditions, you can see that there is a significantly less migration of the sperm throughout, like up the reproductive tract through the cervix. We looked on the DNA fragmentation. So DNA fragmentation within the sperm is associated with lower conception. When we are looking at the higher risk of miscarriage and lower rate of preterm birth. And when we looked, we saw that there is significantly higher DNA fragmentation rate within the dysbiotic condition in the chip. Next, we looked on a hyaluronic binding assay. So hyaluronic acid is the main extracellular matrix within the cumulus. So the cumulus, those are the cells that surround the oocyte, and the sperm has to bind to the hyaluronic acid in order to reach and fertilize the egg. So we saw that there was significant reduction in the ability of the sperm binding hyaluronic acid. So then we'll see whether, if we will treat the patients, whether it can help. So we designed the cervix chip with dysbiotic condition, and we treated with the common known antibiotics that is known to be effective against this condition. We compared it to live bacteria product. So this is in combination of 13 strains of lactobacilli that were shown before that they have the ability to thrive within the female reproductive tract and within our chips. Okay, so first we looked how the host is responding to the different microbiome conditions and how it responds to the treatments. So we looked on the epithelial cells, and we saw that despite the fact that we are treating with a significant amount of conditions, our epithelial cells thriving well. We look on clue-like cells. So clue cells are the hallmark for dysbiosis, for bacterial vaginosis in a clinical settings. So those are epithelial cells that are covered with bacteria. And we saw that when we are exposing to the chips to the dysbiotic condition, we have a significant amount of clue cells, and when we're treating them with antibiotics, with metronidazole or live therapeutic product or both, we have significant reduction in the clue cell. Next, we looked how the host is responding. We looked on the cytokine production, and we saw that there is significant reduction with the cytokine production in our treatment groups. And then we looked on sperm function. So we look at the DNA fragmentation, as I said before. So clinically, we evaluate sperm for fragmentation and to see whether we can, this is something we know that can well show what will be our chances for fertility. And we saw that when we are treating, when the sperm is introduced to the dysbiotic condition, there is a significant amount of DNA fragmentation, and following treatment, we see the reduction. Next, we looked on hyaluronic acid, a binding assay. So basically, you can see here, this is a plate which is covered with hyaluronic acid, and we introduce sperm, and we see what is the percent of sperm that has the ability to bind compared to those that cannot. And we saw that there is significant reduction in dysbiotic condition and an increase after following treatments. Oops. So basically, I think the first thing we saw, we showed that that organ on a chip is a good model to study female reproductive tract dysbiotic condition. We have the ability to evaluate the interaction between the female reproductive tract, the dysbiosis, and the sperm function. We saw the dysbiotic condition, they affect the ability of the sperm to move up the reproductive tract. They can affect the DNA fragmentation. They can affect the binding to the outside. Yes, and with this, I would like to thank the team that took a significant a part of this work. And I am back to Israel now, and I'm starting my lab, and we are working in organ on chip, and we are working on dysbiotic, and we have few different paths that we are going to, and I will be super happy to collaborate. So if you have anything that would be interesting, I will be super happy to collaborate.
Thank you very much, Ola. That was great. Any questions?
Yeah, I would like to ask something. Great talk, Ola. Thank you. So you say that you're introducing this cocktail, and I was wondering, maybe I missed it, but do you have a profile of the outcome of the microbiome that is in the chip after this introduction?
Yeah, it's a great question. So yes, so I presented like a piece of evidence that is that how we are translating this work, but basically the preliminary result of the work was showing that we have the ability to introduce the bacteria, to grow them for a while, and following this, we did like RNA-seq and showed that still the dysbiotic condition are still there. So we are evaluated first just a combination of commercial available bacteria, and then we moved to clinical samples of patients with dysbiosis, and we showed that following the introduction to the chip, we are maintaining the relative abundance of the bacteria.
Abigail.
Yes, hi, sorry. Thank you. Is there anything that you find between the different dysbiotic microbiomes that cause this effect that's in common, that you could sort of have a clue to what is causing this?
The dysbiotic condition?
What about because I assume dysbiosis is a very general thing. Is there specific bacteria that's overtaking? Are they changing the pH? I don't know. Is there something that you could mechanistically say about it?
So yeah, so the dysbiotic condition, we have like 5 types of dysbiotic condition. The one that we use here is bacterial vaginosis-like condition. So it changes significantly the environment. It increases the pH. It's increased the production of cytokines. It disturbs the barrier of the epithelial cells. It creates more clue-like cells. Yeah, and we showed all of it in previous works. And yeah, I cannot go back, but the first slide that I just briefly went through, this is something that was just published 2 weeks ago in Nature Women's Health. So we showed how the cervical mucus affects the vaginal environment of the microbiome, how it affects, and yeah, it has a significant effect.
Okay, thank you, Ola. In the interest of time, I think we need to move forward. So thank you very much.
Thank you so much.